Hey guys, today I'll be discussing dot point 2.1 from the topic area 2 of Blueprint of Life, which is in the HSC biology syllabus. It requires us to actually explore the experiments of Gregor Mendel. Now, Gregor Mendel is quite an inspiration. He's an absolutely amazing scientist, and you should really learn what he did to understand this topic. And to do that, we have to look into his experiments. Gregor Mendel, I'm just going to go a little bit into a historical outline and what he actually did. Which isn't written down on this page. Gregor Mendel in 1850s he created an experiment to figure out how inheritance works. Now at the time he didn't know that there was such thing as DNA, and he didn't know that we had these matching genes. And it's only because of him that we were able to figure out how these genes worked. Okay, so starting off, in the 1800s, Gregor Mendel grew up in quite a poor family. He wasn't actually that wealthy, and nor was his family. They ran a farm, and they actually realized how smart Gregor Mendel was, and ended up paying for him to go to a private school, or a school for him to expand his knowledge. He ended up going to the school, expanded his knowledge, and then over a tragic incident, his father became disabled on the farm. As traditions are, normally the fathers hand down the farm business down to their sons, and Gregor Mendel decided that he did not want to do it. He actually wrote in a diary that, hey, this is too exhausting for me, I can no longer stay on this farm, I am leaving to go to the nearest monastery. So what happened is that this Augustine person, Gregor Mendel, ended up going to a monastery and became a monk. For the rest of his life he was considered a monk, and that's where his great experiments were. So let me just start about the experiments. In around 1856 or near that time, he started his experiments, and these experiments were based off peas. Yes, that might sound weird, but he actually figured out how inheritance works through the characteristics passed down in peas. So let's have a look at what he did. Just a quick outline would be that he worked with pure breeding plants. The key word is pure breeding. And then hybrids. Mendel crossbred tall and short plants. All of the offspring were tall. He allowed all of these offspring, which is the first generation, to fertilize, and found that the F2 generation consisted of tall and short plants in the ratio of 3:1. There's also another important ratio that you must remember. It's called the Monohybrid Ratio. In this topic area, you will learn a bit more detail about this monohybrid ratio, but just getting introduced to it, 3:1 is actually a pretty interesting ratio that comes up a lot in his experiments. And then he repeated this experiments, testing other characteristics of garden peas. So he tested stuff like whether or not the seed was round or wrinkled, whether it was yellow or green, smooth or constricted pod, and yellow/green pod, whether if it was a violet or white flower, tall or short stem, terminal/auxiliary flowers, which basically means top or along the stem. Now these things that he tested, they were incredibly successful, and you'll find out why in the next dot point, and pretty much my next video. But going on, let's go into even more detail. I know this is only an outline question, but if you know more about it, the rest of the syllabus and the rest of this topic area 2 will be much easier to understand. Okay, so what actually happened? Darwin and Wallace proposed their theory of evolution, but they had no clue about the mechanism for inheritance of these characteristics. Gregor Mendel, as I said before, he's considered the founder of modern study of genetics, which is also known as heredity, and he was an Austrian monk. In 1856, he carried out experiments to study the genetics of garden pea plant, which is also scientifically called Pisum sativum, and how certain characteristics were inherited from one generation to another. So Mendel's experiments actually consisted of something close to this. He selectively bred plants for each characteristics for 2 years. As I said before, something you should remember, he bred them for over 2 years, and that's quite dedication that I really like about Gregor Mendel. And he only produced pure breeding offspring. This was mainly because he wanted to make sure that the experiment was valid, reliable, something that was also accurate. It's what led him to the success in this experiment. He then performed cross-pollination experiments with the pea plants that differed in one trait, for example, pod color. So what I mean by cross-pollination is that he'd actually get the go to the anthers of one pea plant and manually transfer the pollen from one pea plant to the other in order to allow cross-pollination to occur, and hence that way he was able to control which plants bred with which, and he was able to track the characteristics that the offspring produced. Mendel chose 7 pairs of characteristics that he wanted to study. As we said, we looked at these before, so you don't really need to worry about it. You can just read over it. And he did this, as I said before, once again, that he manually transferred the anthers, right, of each pea plant in order to do what he wanted, in order to breed the specific plants that he wanted to breed to see the outcomes. And from these outcomes, these two, firstly, crossed two pure breeding plants, then crossed the offspring, and that way Mendel put forward his laws. He had 2 real laws that he wanted to propose. His 2 laws were that random segregation, which is each pair of homologous chromosome is sorted independently during meiosis in sex cells. Independent assortment, which was the principle that during meiosis 2 copies of each genes are created then distributed to the sex cells independently of the distribution of genes. This, I don't think you need to remember that much, but I would say remember everything else from that point above. So I'd just say remember his experiment, remember what he did, remember what he tested, not everything, but you can just remember a few characteristics which will help you inside a test, or suppose an essay where you wanted to discuss what he tested, and also how he did it, which was through manually transferring pollen from the anthers, male part of the plant, of one pure breeding plant, to a contrasting pure breeding plant. But he removed the anthers from the contrasting pure breeding plant so that plants did not self-fertilize. Okay, that's pretty much it for this dot point, and I really do hope this helps. See you next time.